Hello everyone, today we will be presenting our research on reimagining brain anomaly segmentation and classification using MVFA and ResNET-50. In MRI-based brain anomaly segmentation and classification, existing models often segment only one modality or classify a single tumor type. Our research addresses these limitations by testing two approaches, first using a single modality model to segment specific anomaly regions within whole tumors, and second, implementing multi-class classification with segmented anomaly masks. This work aims to enhance MRI-based anomaly detection by supporting both regional segmentation and multi-class classification. Our goal is to segment and classify brain MRI images with tumors into their corresponding labels using different imaging modalities. For segmentation, we define three regions: enhanced tumor, non-enhancing tumor core, and surrounding flare hyperintensity. For classification, we categorize the tumors into three types: glioma, meningioma, and metastasis. For this project, we used the BRATS Challenge 2023 and 2024 datasets with 369 by 369 pixel MRI images for three tumor types: glioma, which are malignant tumors in the brain or spine; meningioma, which are benign tumors in the meninges; lastly, metastasis, which are the tumors that have spread from other body parts.
Each dataset includes 4 MRI modalities and their corresponding segmented ground truth images. The 4 modalities include T1-weighted non-contrast, which are useful for general brain anatomy; T1-weighted post-contrast are to highlight abnormal areas; T2-weighted for fluid detection; and flares highlighting tissue abnormalities by suppressing fluid signals. These modalities are used to help visualize different tumor characteristics, aiding in the segmentation of ET and NETC and SNFH regions. For ET, we have overlaid T1C and T2W modalities. The T1C modalities was used for NETC, and overlaid T2W and T2F modalities for SNFH. For our methodology, we utilize two pre-trained models: MVFA for anomaly segmentation and ResNET-50 for classification. First, we utilize the multi-level visual feature adapter, or MVFA, which adapts a visual language model like GLIP, which is used for medical anomaly detection. We used a 16-shot training approach to avoid issues with NAN values. Training that was done over 50 epochs, selecting high-quality images to optimize ET and ETC and SNFH segmentation. We also applied the Otsu thresholding method to refine the heat map segmentation results. For the second model, we have implemented ResNET-50, which is pre-trained on ImageNet for the classification of 3 brain tumors. We have split our segmented result into 80% training, 10% validation, and 10% testing. We unfroze the top 10 layer of ResNET-50 and inserted more dense layer with switch activation to adapt to our data. The final softmax layer generates classification probabilities for each tumor type.
Now we will explain the overall framework. Starting with the original BRATS dataset, we took one slice from the center of each modality to obtain the 2D image of the brain. These images are then overlaid with the required modalities for easier detection on the corresponding tumor region. The original ground truth images were also further split into 3 images via color value filtering on grayscale. We then trained and tested them with the MVFA model, obtaining the heat map, and converted it to our segmentation results. After obtaining the segmentation results from MVFA, we overlaid them into RGB images, with red for ET, blue for NETC, and green for SNFH. These results were then trained and tested through the ResNET-50 model in order to classify them into the 3 brain tumors: glioma, meningioma, and metastasis. Here are the segmentation results from MVFA for each of the 3 tumor types. This shows our combined segmented anomaly results in comparison with the ground truth and original brain images. The segmentation accuracy is high, with 98 to 99% for the pAUC and above 87% for the AUC of ET, NETC, and SNFH. This shows MVFA's effectiveness in visualizing various tumor regions accurately. Using ResNET-50, we achieved a test accuracy of 68.24% and a test loss of 2.99, with challenges mainly from limited data and unrecognizable features. In our research, we encountered several limitations, namely limited feature recognition due to the small dataset imbalance, MVFA focusing on single modal images, potentially losing information from overlaying multiple modalities. Tiny tumors are hard to detect due to their subtle appearances. And lastly, only the center slice is taken, and we did not try all possible slices from the 3D brain. To overcome these challenges, we plan to 1. Increase the dataset size for better classification results. 2. Integrate more tumor features if additional datasets are available. 3. Fine-tune MVFA or explore other model options tailored to medical imaging. And 4. Experiment on all possible sizes. In conclusion, our approach demonstrates the potential for combining segmentation and classification models to improve brain tumor analysis in MRI scans. MVFA showed promising results in anomaly detection, though ResNET-50 requires more robust data to achieve its full potential. Thank you for listening.